So, good evening dear students. So, today we will be discussing a very nice topic and that topic is our immunology and how defense mechanism is maintained inside our body. That topic we will discuss today. So, any bacteria, any virus, if it is affecting our body, so our body has its own defense mechanism to counter the bacteria and virus. So, today we will be discussing by giving one example of throat infection. So, how the defense mechanism is happening inside our body, that I will discuss in front of you. Okay. So, let me discuss. Suppose this is our body, this is our face, this is, we cannot say that it is a male or female, but it is a face. Okay. So, here what is happening, if we are having any kind of bacterial infection over here, so suppose we will take a discussion on throat infection. Okay. So, if a throat infection is happening over here, so what will happen? Now, in case of throat infection, we will find bacteria are here. These are the bacteria we will get over here. Okay. So, when bacteria will get over here, so what will happen to the next thing? When bacteria are here, what will happen to the next thing? Now, bacteria is such a thing, it will release the toxins. So, when bacteria will release the toxins, so what will again happen? So, when bacteria will release the toxins, what will happen? Now, these toxins so are you getting my point what I am saying? When bacteria is coming to your throat cavity, so suppose this is your throat cavity, okay, this is your throat cavity, and when bacteria is entering into your throat, okay, very clearly I will discuss in front of you. Okay. So, suppose this is your throat cavity, through the nasooral route, if bacteria is coming to you, through the nasooral route, if bacteria is coming to you, so what will happen? Now, suppose this is one bacteria coming, this is a bacteria, it is found in the throat cavity. Okay. So, this bacteria, as you know, the bacteria is having toxins, and these toxins will start damaging the tissue of that part. When the bacteria will release the toxins and these toxins will damage the tissue of that part, how it will damage the tissue of that part? A single bacteria can cause minimal damage, but when the bacteria will be in multiple number, it will create more damage to the tissue, tissue of the throat. So, when a bacteria is coming to the throat cavity, so instantly what is happening? Now, that bacteria is becoming multiple in number. When bacteria is becoming multiple in number, what is happening there? Now, this bacteria will give more number of, or you may say more toxins. Okay. So, more tissue damage. So, first of all, bacteria becoming what? Multiple in numbers. Okay. Are you getting my point? So, when bacteria is throat infection, then in the mucosa you will get bacteria, then bacteria will be multiple in number, then multiple bacteria will release toxins, that toxins will cause underlying tissue damage. So, this tissue is getting damaged and also that will cause underlying tissue damage. Here you may say this is your underlying tissue damage, and this is the wall of the pharynx, this is the wall of the pharynx, where what will happen? These bacteria will release toxins over here. Okay. So, what I am discussing, I think you are understanding. Again I am repeating it. That when you are affected by any bacteria, virus or any other organism, okay, whenever you are affected by bacteria, virus or any other organism, what is happening? Yes, please. Whenever you are affected by any bacteria, virus or any other organism, what is happening to you? Now this bacteria will come to the throat cavity, okay. When the bacteria will come to the throat, then what will happen? The bacteria will be present in the mucosa of the throat, then bacteria will be in multiple in number. When bacteria will be in multiple number, they will create tissue injury, they will do tissue injury because they will release the toxins. Then that bacteria will cause damage to the underlying tissue, and this bacteria will cause damage to the underlying tissue, okay, and this bacteria will release the toxins and these toxins will damage the underlying tissue. So why I am discussing all these things to all of you? Because I am discussing all these things to all of you because you see many questions are coming from the immunology chapter, that by question wise I will discuss all these things. So this is the bacteria, multiple in number, damaging the throat tissue, then bacteria are releasing again toxins. When toxins are released from the bacteria, these are the toxins released from the bacteria, you see these are the toxins released from the bacteria, okay. Now when toxins are coming to the underlying tissue, then these bacteria are also causing what? They are causing damaging of the what? They are causing damaging of the pharynx. These bacteria are again coming to the wall of the pharynx, they are damaging the pharynx, okay. There are three parts of the pharynx, you know, parts of pharynx, oropharynx, nasopharynx, these are asked in the examinations. Okay, so I am not going into detail of it. When I will discuss anatomy, I will come to it. So bacterial is now coming to the pharynx, so pharyngitis is happening. So where an acute phase of reaction is happening, acute phase of inflammation is happening. As you people know that when acute phase of inflammation is happening there, so what will happen? So when inflammation will happen, so there is a series of processes are there in inflammation, I will discuss in surgery context, but now I am saying when inflammation is there, so there will be vasodilatation. Why? Because these chemicals will act on that vascular area of the tissue, and tissue will be damaged, there will be vasodilatation. So what will happen if there is vasodilatation? So particularly arterioles are getting dilated. So when there will be vasodilatation, what will happen? Let me explain you. Suppose in this area I am erasing the toxins, don't worry, green color are the toxins, and in this area suppose there is vasodilatation. So if the blood vessels is dilated over here, okay, this is the area of the pharynx, don't worry. Now in the bacteria we are in the throat cavity damage, and the bacteria become multiple in number, then they release toxins, toxins coming to underlying tissue. This is underlying tissue, pharynx, okay. So this is your underlying tissue, again I am confirming it, okay. This is underlying tissue, pharynx. When bacteria are coming to the pharynx, okay, so pharyngitis is happening and they are also multiple in number, bacteria are becoming multiple in number. Now you will get bacteria are multiple in number over pharynx, okay. Here you will get multiple number of bacteria, okay. So now is it clear to all of you, my dear students? So you can put your query in chat box if you are having any doubt. So number one point, again I am saying it is a chapter of immunology, then I am discussing about the immunological responses of our body, and which are basically done by the WBC, the white blood cells, and you will be getting neutrophil, lymphocyte, monocyte, all these things in white blood cells. So when bacteria is entering into our throat cavity through the nasal route, and in the throat with the mucosa, bacteria are getting multiple in number, they are releasing toxins, there will be tissue damage, and these bacteria will again cause the underlying tissue damage. This is the underlying tissue, and in the underlying tissue damage, these bacteria are coming to the wall of the pharynx, then they are damaging the pharynx, there will be pharyngitis, and where there will you will get multiple number of bacteria. This is all about bacteria entry into your throat cavity and also pharynx. Multiple number of bacteria will happen, or multiple number of bacteria will get, okay. So next we will come to another point is what? When bacteria will be in multiple number in the pharynx, so what will happen? There will be an acute phase of vasodilatation there. Why vasodilatation there? Because you know that when there will be any type of infection, any type of tissue damage, so what will happen? There will be vasodilatation over their area, blood vessels will be dilated. It means particularly capillaries will be dilated, arterioles will be dilated. So when the blood vessels will be dilated, what will happen? when the blood vessels will be dilated, what will happen? When the blood vessels will be dilated, my dear friends, suppose this is your blood vessels, okay. When the blood vessel is getting dilated, what will happen? So there will be more flow of blood, okay. So when blood vessels will be dilated, there will be flow of blood, okay. So when there will be more flow of blood to the blood vessels, then again what will happen? All the defense mechanism maintained by your WBC, yani the defense mechanism maintained by your neutrophil, okay, maintained by your monocyte, maintained by your lymphocyte, maintained by your basophils, all the cells are there, na? They will start flowing over the blood, over the circulation, and these neutrophil, monocyte, lymphocyte, basophil, they will come to the active sites here, they will come to these sites. Why? Because they are the defense mechanism system inside our body, and they will kill the bacteria. Not only neutrophil, monocyte, not only the WBC, not only neutrophil, monocyte, lymphocyte, basophil, but other antibodies will also come to this place, okay, or many other things will also come, will come through the circulation. So my concern is right now what? After the tissue damage, there will be vasodilatation, and particularly the dilatation of the arterioles. So keep it in your mind, vasodilatation, particularly what? Sorry, particularly the arterioles. Particularly the arterioles, okay. So in the vasodilatation, what will happen? There will be flow of the WBCs to the circulation. Then in the WBC, neutrophil will be coming, monocyte will be coming, lymphocyte will be coming, all the things will be coming. I am repeatedly saying it because it is very important. Okay, then what will happen? You see, then where in this situation, inside the blood vessels, a very special type of mechanism is happening. As you know, our blood vessels internal lining, these are the endothelial lining. These are the endothelial lining. So in this very moment, what is happening? From the endothelial lining, a special type of event is happening. That event is what? But a secretion of a sticky material. So during this vasodilatation process, what is happening? There will be a secretion of some sticky materials. Some sticky materials are secreted from the endothelial lining of the blood vessels. Are you getting my point? So what is the intention of it? Entrance question. It is an entrance question. During the acute phase of infections, vasodilatation will be there, and in the vasodilatation, what is secreted from the endothelial lining, internal lining of the blood vessels? That one is nothing but a typical sticky material. Typical sticky material. What is the intention of secretion of the sticky material? Why? Because whenever neutrophil is coming, you see, whenever, suppose this is your neutrophil, whenever your neutrophil is coming, this is your neutrophil, multilobed. Whenever your neutrophil is coming, okay, and whenever your monocyte is coming, okay, whenever a neutrophil is coming, whenever your monocyte will be coming, suppose this is your monocyte is coming, okay, whenever neutrophil is coming, whenever monocyte is coming, at that moment these sticky materials will help the neutrophil and monocyte to stick it to the blood vessels. It means to stick it to the site of the blood vessels where there are multiple number of bacteria and where there are infections. Are you getting my point? If you are not understanding my point, just put in the chat box that what you meant to say, okay. So the intention of the secretion of the sticky material is nothing but to attract this kind of neutrophil and monocytes and to keep to stick the neutrophil and monocyte to the blood vessel site, and these blood vessels we are particularly dilated where there was acute infections. Got my point? So now the question is coming that the first, when these type of things are happening, the first thing which is coming to the dilated blood vessel, that one is which one? It is a question to all of you. Why, when there is acute reaction, which amongst the WBC comes first to the reaction side or comes first to the dilated blood vessels? My question to you, and you will answer it. So I am giving you the answer. The first thing that is coming to your first WBC that is coming, that one is your what? Yes please. That one is your what? That one is your neutrophil. Okay. So what is the reason behind why neutrophil is coming? So it's an entrance question normally asked to you. The first line of defense mechanism. First line of defense mechanism. The first line of defense mechanism is maintained by, always asked in the examination, that one is a neutrophil. The first line of defense mechanism maintained by neutrophil. Why neutrophil? Because neutrophils are abundant in number. There are more number of neutrophils are there, that's why they come first. Second point is what? Neutrophils are what? More in number. Neutrophils are more in number. And what is the reason why neutrophils come first? The second cause is what? Neutrophils are more what? More motile. So first thing is why WBC are coming? Because during acute phase infection, blood vessels are dilated. The internal lining of the blood vessels are secreting a special sticky material, and these sticky materials are holding the neutrophil and monocytes which are coming out to the sides. The first thing. Second thing is why neutrophil coming first? Neutrophil is the first line of defense mechanism. Why it is coming? Because there are more number of neutrophils. It means neutrophils are abundant in number. Second thing is neutrophils are very motile in comparison to other WBC. And third cause is what? The neutrophil is having chemotactic agents to attract the neutrophils are produced most early. So very important thing is what? Some chemotactic agents are there. The third thing is what? Some chemotactic agents are there. Some chemotactic agents are there. Okay. Some chemotactic agents are there which are secreted in the first phase of reactions to attract the neutrophil, and they are produced most early. It is very important. They are produced what? Most early. So it is very important, my dear friends. So kindly keep it in your mind that when there is acute phase of reactions, why neutrophil comes first. Number one point, it is more in number. Number two, it is very motile in nature. Number three, some chemotactic agents which are most early secreted, and as a result of which neutrophil is coming first. Okay, getting my point? So now one question we got from our discussion, that is first line of defense mechanism is maintained by neutrophil. So why neutrophil and monocytes are coming to the point and getting stuck there? Because the internal lining of the blood vessel is secreting special sticky material. Okay, so these things we got important from the entrance point of view. Next we will discuss another thing. That one is your very important thing, that the after the first line of defense mechanism we will discuss about second line of defense mechanism. So which one is there which is exactly maintain the second line of defense mechanism? Okay, so this was first line. The next is second line of defense mechanism. Put the answer in the chat box. Second line of defense, which one? Answer is which one? Monocyte. Acha, can you tell me what is the difference between monocyte and macrophage? It is very interesting and normally asked in the question in the examination, what is the basic difference between the monocyte and macrophage? My dear students, you remember one thing, that when monocyte is in the blood vessel, that will be called as monocyte. When monocyte will come to the active site to counter the bacteria, that will be called as macrophage. Mean to say that it was your neutrophil, you see, it was your neutrophil. So now neutrophil released, and that will come to the bacterial side, and that will act on the bacteria, and that will try to damage the bacteria. How neutrophil will damage, that I will discuss, don't worry. Then after the neutrophilic action, then there will be coming up what? Commencing of the what? Monocyte. So monocyte, when monocyte is here, that is called as monocyte. Getting my point? When monocyte is coming from inside, it means when monocyte is coming from the blood vessels to the bacterial sites, you know, suppose this one is M for monocyte, and the green one is N for neutrophil. So neutrophils are acting on the purple colors, means these are the bacteria, and after the neutrophilic action then our monocyte is working, and monocyte is yellow marking I have given, and these are also working on the purple color bacteria. Okay, so monocyte is the second line of defense mechanism. Why monocyte is second line of defense mechanism, and what is the difference between monocyte and macrophage? So again I am saying, until monocyte, when it is in the blood vessel it is monocyte. Monocyte, when it is coming out of the blood vessels it is called as macrophage. Getting my point? So always you remember one thing, that neutrophil will start acting on the bacteria, neutrophil will start killing the bacteria, followed by macrophagic action, or followed by macrophage coming. So that's why first line of defense mechanism in our body is neutrophil, second line of defense mechanism is our maintained by our what? Macrophage. Okay, so this is the second question from entrance point of view. Next is what? Neutrophil is multi-lobed. It's a very important question. Neutrophil is multi-lobed. It is coming in the entrance examinations. Multi-lobe. Neutrophil is what? Yes, please. Multi-lobe. Neutrophil is multi-lobed, and neutrophil is otherwise called as what? Microphage. Neutrophil is otherwise called as, yes please, microphage. So many questions on neutrophil. First line of defense mechanism maintained by neutrophil. Why neutrophil coming first? Number 1, the neutrophil is more in number. Number 2, neutrophil is very motile. Number 3, chemotactic agent most early it is secreted. So that's why neutrophil first. Then followed by monocyte secretion. So what is the difference between monocyte and macrophage? When monocyte is inside the blood vessel it is called as monocyte. When monocyte is coming out of the blood vessels to the active site of the bacterial infection, that is called as the macrophage. So neutrophil is otherwise called as what? Microphage. Monocyte is called as macrophage. Neutrophil is multi-lobed. That I have already. This is neutrophil. This is multi-lobed. 1 lobe, 2 lobe, 3 lobe, multi-lobed. But your monocyte is what? Your monocyte is irregular body, and also that is kidney-shaped nucleus. So here nucleus multi-lobed and monocyte is what? Kidney-shaped nucleus. I have already given the diagram. You see, this is irregular body surface. Monocyte irregular body surface, and the nucleus is what? Kidney-shaped. You see, nucleus is kidney-shaped. Okay. So second line of defense mechanism is maintained by macrophage, monocyte macrophage, and macrophage is the big eater. What is the other name of the macrophage? Big eater. What is the other name of macrophage? Yes please, big eater. And what is the nucleus is what? Kidney-shaped. Nucleus is kidney-shaped. And monocyte inside the blood vessel it is monocyte. Monocyte outside the blood vessel it is macrophage. Hope you have understood what I told. Okay, then next is what? Then next is, we will come how neutrophil damage the bacteria. This is most important. Then come to this point, when neutrophils are coming to the active bacterial infection site, then how neutrophils are killing the bacteria. It is very easy how neutrophils are killing the bacteria. Let me explain you. Okay, how neutrophils are killing the bacteria. So as you know, that it's the process of phagocytosis. When neutrophil is coming, when neutrophil is coming to the active bacterial side, infection side, then instantly what is happening? Then instantly your neutrophil is engulfing the bacteria. The neutrophil is engulfing the bacteria, then some special type of chemicals are released. So when this special type of chemicals are released, so then neutrophils are killing the bacteria over there. Okay, so let me discuss about, let me draw the diagram again. It was very distressing. Okay, so next coming to the point, that this is a man. Okay, this is a man like you people. Okay, so here there are bacterial infections. Okay, again I am drawing. Here there are bacterial infections, multiple in number, then toxin release, then tissue damage, then these bacteria are coming to the, you mean these bacteria are coming to the underlying tissue, okay, these bacteria are coming to the, let me draw the diagram again. So diagram is very important to be drawn very rightly, and also diagram should be looking very nicely, okay. So I am becoming acquainted with the board. So let me draw the diagram again. This is a very beautiful person, okay. Okay, this is a very beautiful person where there are bacterial infections. Again and again I am giving the diagram of bacterial infections. So where this is the throat cavity, bacteria is coming with the mucus. So these are the bacterial infections, okay. Bacteria are now multiple in number. When bacteria are multiple in number, they are again creating tissue damage, okay. When bacteria are doing tissue damage, then underlying tissue is getting damaged, and this is the underlying tissue, you see, this is the underlying tissue damage, this is the underlying tissue damage, and this one is the pharynx, and bacteria are again coming to the pharynx, bacteria are again coming to the pharynx, okay. So bacteria are again coming to the pharynx, okay, bacteria are again coming to the pharynx. Then there will be blood vessel dilatation. I have already told blood vessel dilatation. First line of defense mechanism is neutrophil, then second line is the monocyte, already told monocyte, then neutrophil will come to the site, then monocyte will also come to the site. So these are the things happening over here when any person is affected by any throat infection. Then let me discuss what is happening in the neutrophil when bacteria are coming to it. So in the neutrophil, what is happening? Neutrophil will, you see, let me draw a neutrophil. Suppose this is your neutrophil, okay, this is your neutrophil, and neutrophil I have already told that neutrophil is multi-looped. I have already told that neutrophil is multi-looped, okay. So this is neutrophil, it is multi-looped. So what is happening when neutrophil is coming to the site, bacteria site? So when neutrophil is coming to the site, it will secrete destructive enzymes. The neutrophils will secrete the destructive enzymes. So if it is a neutrophil and if it is a bacteria, you see, if it is a neutrophil and if it is a, suppose this is your bacteria, okay, this is your bacteria, and if it is a neutrophil, then neutrophil is doing what? Then neutrophil is secreting some destructive enzymes. It is very important. Note it down. Some destructive enzymes. Okay. Neutrophil is secreting some destructive enzymes. So what is happening when destructive enzymes are getting secreted? Now how it is doing? Now you see, in the neutrophil, let me make a concise diagram of neutrophil. I am making it more in, big in size. Suppose this is a neutrophil, okay, this is a neutrophil, okay, this is multi-lobed, and the neutrophil has already engulfed the bacteria. And what is happening inside the neutrophil? That in the neutrophil there is enzymes will be, lysosomal enzymes will be produced. How the lysosomal enzymes will be produced? You see, so there will be enzyme productions will be in this manner. Destructive enzymes, these are destructive enzymes. These will be produced in this way, in this manner. Okay, this enzyme. Okay. Okay. So these destructive enzymes will be secreted from the neutrophil. These are the pink color दे देंगे, ठीक है? तो destructive enzymes will be secreted. These are the enzymes will be secreted from the neutrophil. Okay. Then these enzymes will be secreted, and these enzymes will act on the bacterial side. Okay. These enzymes will act on the bacterial side. Then let me discuss with you another one thing, that On the bacterial side, there are antigens. You know antigens, every bacteria has antigens, and the antigens are of different types. You mean you cannot get same type of antigen in one bacteria. In one bacteria, you will get different type of antigens. Another one, entrance question. That one is same bacteria. Entrance question. Same bacteria contains which of the following antigens, type of antigens? Same bacteria contains what? Different kind of antigens. Same bacteria contains different kind of antigens. Okay. So, like I am giving example of antigens over the bacteria, you may say, suppose this is one kind of antigen. You may say this is one kind of antigen. This is one kind of antigen. Okay, and this is one kind of antigen. You may say, so one bacteria may contain different kind of antigen. So what is happening? So when the neutrophil is coming, let me draw the neutrophil this color, it will be easy. So this one is a neutrophil. When the neutrophil is coming to the site and acting on the bacteria, so this neutrophil is doing what? This neutrophil is engulfing the bacteria, and around the bacteria you will get different kind of antigens, like this is star antigen, this is round antigen, like this antigens. And this neutrophil will secrete some lysosomal enzymes. So these lysosomal enzymes will kill the bacteria. So when the lysosomal enzymes will kill the bacteria, then at last what will happen? Bacteria will be damaged. My dear friends, bacteria will be damaged. Okay, bacteria will be damaged. You see, so when bacteria will be damaged, remaining will be what? When bacteria will be damaged, remaining will be what? Remaining will be only what? Antigens. So when bacteria are getting damaged, but remaining is what? Antigens. You will get these antigens. So what will happen to the antigens which are freely found in the neutrophil? Now what will happen? These antigens, these free antigens will now drain to the lymphoid organs. You mean these antigens will come to the lymphoid headquarters. We have many lymphoid headquarters, so these antigens will come to the lymphoid headquarters, and these antigens drain through the lymphatics and when down to the lymph nodes where there are lymphocytes. So the main thing is what? The free antigens now, what I told, the free antigens now drain through the lymphatics and somewhere down to the lymph nodes where there are lymphocytes. Okay, getting my point? So now I will write, let me write, these antigens will, write it, note it down, these antigens drain, okay, to the lymphatics. These antigens drain to the lymphatics and then कहाँ जाते हैं वो? lymph nodes. Okay, then कहाँ जाते हैं? lymph nodes. इसके बाद कहाँ जाते हैं? lymph nodes जाते हैं, और where there are what? They will move down to lymph nodes where there are lymphocytes. और lymph nodes में क्या क्या होता है? Lymphocytes होता है. I think you people are understanding. Okay. So why it is so important I am discussing? You see one thing, that when neutrophils are working on it, neutrophils will engulf the bacteria. Neutrophils, I have already told, this is multi-lobed nucleus. Then when neutrophils are engulfing it, neutrophils are releasing some enzymes, pink color of destructive enzymes. Then bacteria will be destroyed. When bacteria will be destroyed, there will be free antigens. So again I am saying one question, same bacteria contains different kind of antigens. Okay, same bacteria never contains same type of antigens. So these antigens will drain to the lymphatics and finally lymphatics, then again it will be drained to the lymph nodes, where in the lymph nodes you will get the lymphocytes. So this is all about the neutrophil, but the very interesting thing is that what is happening to the monocyte, and that is very interesting to know. Neutrophil's work is complete here. The free antigens will come to the lymphatics, then in the lymphatics, then to the lymphocyte, then its activity is over here. But the main thing is what I meant to discuss here, monocyte. It is very important. What is happening to the monocyte? When monocyte is working, after the neutrophil, monocyte is when monocyte is working, what will happen? And PG entrance question is coming from that part. So let me discuss about the monocyte. What is happening to the monocyte? When monocyte as a second line of defense mechanism is coming, what is happening to it? Okay, so my dear students, put in your chat box that first line of defense mechanism, which one? Neutrophil. Second line of defense mechanism maintained by which one? Which one? Second line of defense mechanism maintained by which one? Monocyte. What is the difference between monocyte and macrophage? Monocyte when blood within the blood vessel, when outside the blood vessel is macrophage. So why neutrophil come first? Because chemotaxis agent release early, it is more in number, it is very motile in nature, okay. Then neutrophil is multi-lobed, it is otherwise called as macrophage. Then monocyte is called as what? Macrophage. Why? It is big eater and it is second line of defense mechanism. Then what is happening when monocyte is acting on the part of the throat cavity? Let me draw a diagram again on it, okay. So when monocyte is coming, you see, suppose this is a beautiful person, okay, whose throat has been infected. This is a beautiful person whose throat has been, let me draw a small diagram on it because I have to cover a lot of things over here. Make it a small diagram. Now you are understanding, so it will be better to draw a small diagram. I think it is visible to all of you. Okay. So this is a beautiful person like you people. So bacteria is infecting you. You know everywhere you will get bacteria. The world is full of bacteria. Bacteria is always coming and infecting us. Okay. So I think there is no need to describe again what is happening. Throat cavity infection is there. Throat cavity infection is there. Multiple number of bacteria again. Okay, multiple number of bacteria. Then these bacteria are damaging the underlying tissue. This is your underlying tissue. It is damaging your underlying tissue, and this damaging your underlying tissue. And this is your pharynx. This is your pharynx. Question आता है anatomy से pharynx कितने प्रकार का होता है? How many types of pharynx are there? Okay, put it in your, put it in answer. Put the answer in the chat box. So these bacteria are toxin releasing toxins. I have already told you, and these bacteria will damage the tissue. Then again, this bacteria will be in multiple number in the pharynx. Then in the multiple number of, when it will be in multiple numbers, then again I have already told there will be blood vessel dilatation. Okay, when there will be blood vessel dilatation, there will be coming of what? Yes, please. There will be coming of, yes, there will be, yes. When there will be blood vessel dilatation, there will be coming of what? Let me draw the blood vessels. First of all, bacteria are coming. So these are the bacteria over here. Then due to multiple number of bacteria, there will be acute phase reactions and there will be inflammatory process will be started. Again in surgery, I will be discussing what is inflammation, how many process of inflammations are there. So here there will be vasodilation. When there will be vasodilation, there will be coming out of, yes please, there will be coming out of, first number one is your neutrophil, then again there will be coming out of, yes please, then again there will be coming out of, monocytes. Okay, then neutrophil, when there will be coming out of neutrophil will act over here, and in my last diagram neutrophilic action was complete. In my last diagram neutrophilic action is coming. Finally neutrophil was coming to what? I have already told. Neutrophil antigens were coming to the lymphatics, somewhere to the lymph nodes where there are lymphocytes. Okay, then after the lymphocytes, then there is coming of what is please? Monocyte. Then when monocyte coming to the bacterial side, there will be called as what? Macrophage. Okay, now let me discuss neutrophil. I have already discussed. Now let me discuss what is happening to the actually monocytes. What is happening inside the monocyte, so that basic concepts of many questions are coming to that point. Okay, so what is happening in the monocyte? So let me discuss, or let me draw a monocyte. This is a monocyte. Okay, I have already told monocyte, irregular border. Okay, so this is a monocyte. So what is happening inside the monocyte, that we need to discuss about. So what is happening in the monocyte? Now we will not discuss it as monocyte, we will say it as what? Macrophage. So what is happening in the macrophage? Now when this monocyte, or now we will say it as macrophage, because it has already come to the site of infection. So now what is happening to the macrophage? Now macrophage now engulfs the bacteria. So when macrophage will engulf the bacteria, same incidents will happen as happened in the neutrophil. So suppose this is a bacteria. Suppose this is a bacteria that has been engulfed by the monocyte, and as I have already told you that bacteria will contain different kind of what? Bacteria will contain different kind of what? Yes please? Antigens. So suppose this bacteria is having this kind of antigen, this kind of antigen, this kind of antigen. Okay, okay, so this bacteria is containing different kind of antigens. Okay, so it has been engulfed by the monocyte. So when it has been engulfed by the monocyte, then monocytic actions will happen over here. So what monocytic action? So monocytic action is this one, you see. Monocytic actions is this one, that I have already drawn in the context of what? I have already drawn in the context of neutrophil. Some enzymes will be secreted, my dear friends. Enzymes will be secreted, and these enzymes will destroy the bacteria. Very simple, I have already told. These enzymes will destroy the bacteria. Very simple. These enzymes will destroy the bacteria. So these are the enzymes which are secreted inside the monocytes. These are the enzymes secreted. Okay, getting my point? These are the enzymes secreted, and these are the destructive enzymes. And these destructive enzymes, a result will come when they will completely what? Destroy the bacteria. So now when the enzymes are getting secreted, you mean yellow enzymes, when yellow enzymes are getting secreted, now bacteria will be damaged. So this bacteria will be not damaged. So when bacteria will be damaged, what will happen? When bacteria will be damaged, what will happen? Now we will get only antigens in the neutrophil. Now we will get only antigens in the neutrophil. Okay. So let me draw a fresh neutrophil. So now let me draw fresh neutrophil, and in the fresh neutrophil we will get what? Yes please, in the fresh neutrophil we will get these are the enzymes secreted. Okay, these are the enzymes which are lysosomal enzymes which are destroying the bacteria. Then now we will found over here only what? We will found only antigens. That I have told that antigens are never destroyed. We cannot destroy the antigens. Antigens will be there. So now the real game will start. Now the real game will start. That what is happening to the macrophage? If I am kindly ignore the term monocyte, now it is macrophage. So what is happening when macrophage is getting the free antigens? What will happen? Neutrophil send the free antigens to the what? Lymph nodes or to the lymphocytes. But now macrophage is never going to sending the free antigens to the lymphatic system, to the lymph nodes or to the lymphocyte. So what macrophage will do? Now macrophage will do some kind of internal episodes, some kind of internal reactions, as a result of which it will process the antigen. So which type of reactions, which type of things will happen inside the monocyte by getting the antigens. So you see that in chromosome number 6 of short arm of both maternal and paternal, there is very special type of gene setup. So you mean, suppose this is in the monocyte. In the monocyte, you know about the chromosome. In the monocyte, what is happening? In the monocyte, in chromosome number 6 of short arm of both paternal and maternal, there is very special type of gene setup and genetic material on top, which always keeps on suppling all times, but genetic material at the bottom remains stable. Let me discuss you. Okay. Don't worry, I will clarify everything. Don't worry. I will clarify everything. So, we all are having, genetic material, we are having chromosome. Okay. So, same thing is happening to the monocyte. In the monocyte, you mean in the chromosome, there is a short arm and there is a long arm. So, in the short arm, so this is a chromosome, okay, short arm and long arm, okay. So, some reactions or something is happening inside the chromosome that is in 6th position. Suppose this is a 6 positions of chromosome. So, in 6th position of the chromosome, it is an entrance question. In 6 positions of the chromosome, so 6 means, obviously, if one chromosome will come to the maternal and second also will come from the paternal region. So, okay. So, 6 positions of the chromosome, what is happening? Now, there will be expression of some genetic, there is expression of, or there is a special type of gene setup and there is expression of some genetic material. Okay. So, there will be expression of some genetic material. So, here in the 6 positions, there will be expression of some genetic material. When there will be expression of some genetic materials, so what will again happen? Let me zoom it. Okay. So, in the 6 positions, when there will be expression of some genetic materials, what will happen? When there will be expression of some genetic materials, we will get what? We will get, I am zooming in this one. So, we will get it like this, you see. I am zooming it like this. So, again I am repeating it, that macrophage is engulfing the bacteria, then macrophage is deriving some enzymes. By the enzymes, bacteria is getting destroyed. Then after the destruction of the bacteria, antigens are getting free. When antigens are getting free, here some genetic expressions are happening. So, in the macrophage at chromosome number 6, a special type of genetic expression is happening, and in the genetic expression, we are finding the expression is like this, where what is happening? One part is like this and this part is like this. So, when this part is like this, so that is called as what? Suppling. This part is suppling part. This part is what? Suppling parts. It is moving. And this part is what? Stable part. This is suppling part and this is stable part. So, again what is happening? What do we get from the suppling part and what do we get from the stable part? From the suppling part, and as you know, this is called as what? Obviously, genetic expression is there, and when genetic expression is there and this type of suppling parts we are getting, it is also important from entrance point of view. Which type of parts are there? One is suppling part, another is stable part. One is suppling, another is stable, and this part is your DNA. So obviously you know that from DNA which one is formed? RNA is formed. So from the DNA, from the this part, from the chromosome number 6 by the genetic expression, you will get what? There will be formations of messenger RNA. Okay, there will be formation of messenger RNA, and when this messenger RNA will be formed from the DNA, and these messenger RNA will be of different kinds. Let me draw. Suppose this is a genetic material DNA, and from DNA messenger RNAs are formed, and these messenger RNAs are of different kind. So as this part was already I have told, this is suppling part unstable, this part is stable part. Then let me draw three kind of chromosomes, like you see, three kind of messenger RNA. So one will be like this, you see, one will be this like this, another will be like this, and the next will be like, it is very interesting, just have concentration on it, another will be like this. So this one is your what? This one is your pink one is your what? It is your fast, it is your fast mRNA, it is your second mRNA, and this is your third mRNA. So getting my point, what is happening in the monocyte? In the monocyte what is happening? Yes please. In the monocyte, chromosome number 6 from both from maternal and paternal, it is expressing a genetic material, and the material is one end is your suppling, second end is stable, and when we zoom it, when we visualize it, when we will show it in big manner, so it will be expressing three kind of mRNA, messenger RNA. One is your first mRNA, second is your second mRNA, and this one is your third mRNA, and this is your DNA, this is your RNA. Obviously RNA is formed from DNA. So this is also a question, it is a very simple question but asked in exam, that RNA is obviously formed, you know that of course from DNA you make RNA, from DNA you make RNA, very important, from DNA we make RNA, it is very important, keep it in your mind forever. Next is what is happening, then these are different from each other and these are remains constant. Can you guess one thing from here? You see that here what is happening, you see this one differ from each other, differ from each other, okay, because it was unstable, it was suppling, so that's why these are differing from each other. Next, the end part, these are remaining constant and these are remaining constant. As you people may be understanding that why Sanjib sir is describing in detail. My dear friends, you remember one thing, what mind doesn't know I cannot see. ठीक है, आपके दिमाग में एक बार ये फीड हो जाएगा 1 घंटा का वीडियो, तो जिंदगी में क्वेश्चन कभी भूल नहीं होगा. इसीलिए आई एम सेइंग वन थिंग, दैट यू जस्ट कीप इट इन योर माइंड फॉर एवर, इट इज वेरी इजी टू अंडरस्टैंड. So these parts are differ from each other and these parts are remaining constant. Then again what is happening? From the mRNA, some special type of molecules are formed and these molecules are nothing but your protein molecules. So suppose these kind of molecules are formed from, you mean from this kind of things, a special kind of protein molecule is formed, that one is your what? A special kind of protein is formed over from here and it will be looking like this, you see. This is a special kind of protein formed from first mRNA, then from second mRNA you will get another kind of different looking protein and that one is like this, you see, from it is very easy to understand. From second mRNA you will be getting this kind of protein, okay, you will be getting this kind of protein. Then from third, then from third mRNA you will be getting this kind of protein. Why I am discussing all these things, I will let you know, don't worry, why I am discussing all these things, everything I will let you know. And you will be astonished to know that sir ऐसे इतना interesting था चीज इसमें. This is again one type of protein. These are formed. Are getting my point? These are again one type of protein formed. Okay. So now come to point that Macrophage, chromosome number 6, genetic expression, suppling part, stable part, DNA, RNA is formed, first mRNA, second mRNA, third mRNA, then these kind of proteins are formed. So now I have shown you only 3 kind of proteins, but remember one thing, thousands of proteins are formed here, thousands of proteins are formed here, and these parts is differ from each other, but this remaining part is constant. So up to this everything is clear, I think. Then what is happening? These protein molecules will be transported to the antigenic side. Every molecule try to fit. Then what is the difference? Why these kind of proteins are formed over here? You see, here antigens were made free, right? Here antigens were made free. So all these things happening in the monocyte only because to make a protein which will be fitted to the antigen. Then the macrophage will start its next activity. You got my point? What I told? I mean to say that here by engulfing the bacteria, the antigens were made free, right? Let me erase these things. By engulfing the bacteria, antigens were made free. Okay. So these were the antigens, like when star antigens were free antigens. Okay, this is one antigen, this is one antigen, these were the antigens over there in the monocytes, macrophage, sorry. So how to counter this, antigens? So to counter the antigens, these entire steps happen. Expression, genetic expression, 6 number of chromosomes, both from maternal and paternal, then suppling end, then this end, then formations of these kind of things, then formations of, then DNA, then from DNA there is RNA, then from the RNA these shapes things were formed, and these are thousands in number. So what is the intention behind why these are formed? These are formed only because they need to be fitted to the free antigen in the macrophage. So now suppose from these mRNAs, from these proteins, from these kind of proteins, suppose one protein is getting fitted to the antigen of the macrophage. Suppose and this you mean this is the antigen getting fitted to the macrophage, and this you mean these type of things or these type of proteins will come to the macrophage. Then again let me draw the same thing. Don't get confused, it is very interesting. Okay. Okay, let me draw the final product. Okay. So these type of things will come to the macrophage. So number one is, first one is, yes please. So this is come to the macrophage. This is coming to the macrophage. Okay. Then next, this one is coming to the macrophage. This one is coming to the macrophage. Okay. Then next is what? This one is coming to the macrophage. You see. Then now you decide which will be fitted to the macrophage. Suppose I am giving you one example. That this will be fitted to this antigen. So instantly what will happen? This antigen will come over to it instantly. Now this antigen will be called as precious antigen. Now this antigen will be called as, yes please, precious antigen. It's a entrance question. What is precious antigens? Precious antigen is nothing but when the antigen is fitted to the protein formed from the messenger RNA. Okay, so this is called as precious antigen, and this precious antigen, now it cannot be inside the macrophage. This precious antigen will come outside of the macrophage. It means it will be on the surface of the macrophage. So now what will happen? Our precious antigen, now our precious antigen, okay, now our precious antigen will come to the surface of the macrophage. Now the precious antigen will come to the surface of the macrophage. Are you understanding, my dear friends? Now the precious antigen processed with the protein or fitted to the protein will now come to the surface of the macrophage. Very good, you are understanding. So so it will come to the, this is called as precious antigen. Then what will happen? Then why I am discussing, why Dr. Sanjib is discussing many things? Why Dr. Sanjib is discussing many things? Then you see, this precious antigen, this is called precious antigen. The precious antigen formed inside the macrophage now it will be coming to the surface of the macrophage. Then what will happen? Now this precious antigen will be coming to the, now this precious antigen will be coming to the lymph node. Now the precious antigen will be coming to the lymph node. Suppose we have many lymph nodes, now the precious antigen will come to the lymph nodes. So here very interesting thing is happening. What? Here very interesting thing is happening here. What? Now the process antigen will come to the lymph node, and in the lymph node you will find the lymphocytes. So in the lymph node you will find the lymphocytes. So the lymphocytes will find over here. What are the lymphocytes you will find over here? These are the what? T lymphocytes. You will find T lymphocytes over here. Okay, you will find T lymphocytes over here. Now the lymphocytes, the process antigen will come to the T lymphocyte, then T lymphocyte will be activated, then T lymphocyte will recognize the process antigen, where the process antigen is properly processed or not. So that's why T lymphocyte has different receptors. So that's why T lymphocytes has different receptors. So the receptors present over the T lymphocytes are what? This is called as, yes please. The receptors present in the T lymphocytes are called as what? T cell receptors. Here very interesting question will be coming in the entrance examination. So here there are T receptors, and these T receptors are called as T cell receptors. Why T? Because lymphocytes are formed from the thymus gland. So that's why T. T lymphocytes have what? T cell receptors. And these T cell receptors will sense the processed antigen, and it will come and fit to it. Now it will come and fit to it, you see. Now it will come and fit to it. I will take only 5 to 10 minutes, then I will cover it, don't worry. Then this is your process, this is your processed antigen, okay. This is your processed antigen. Then this one is your what? Protein formed by the messenger RNA. Protein formed by the messenger RNA. Now it is getting fitted to the T lymphocyte, and it is sensed by the T cell receptor. So here two things are coming, that T cell receptor has two types of sensing part, like on the surface of T cells. On the surface of T cell there are antigen recognizing unit. On the surface of the T cell there are antigen recognizing unit. Antigen recognizing unit means what? Whether the antigen is properly processed or not. So for the antigen recognizing unit is nothing but your what? The antigen recognizing unit is nothing but our TCR. And TCR is your what? Antigen recognizing unit. This is called antigen recognizing unit, or you may say antigen sensor. Very important. Keep it in your mind. Entrance में आएगा. ठीक है. Then next is what? Then on the surface of the T cell, in the surface of the T cells we are getting two things. One is TCR, that is antigen recognizing cell or antigen sensor, and on the surface we are getting one thing, that one is your MSC class 2 molecule, and that one is your what? MSC2 sensor. What? MHC, that is the name of it. What? Yes please. MSC क्लास 2 मॉलिक्यूल. ठीक है. तो हम टी लिम्फोसाइट में दो सेंसर मिलेगा हमको, एक टीसीआर सेंसर मिलेगा, एक एमएससी क्लास 2 सेंसर मिलेगा. व्हाट इज एमएससी क्लास? मेजर हिस्टो कंपैटिबिलिटी सेंसर. दिस मेजर हिस्टो कंपैटिबिलिटी सेंसर, व्हाट इज द वर्क ऑफ इट? दिस मेजर हिस्टो कंपैटिबिलिटी सेंसर recognizing the molecule whether it has been processed with the MSC class 2 protein or not. So the processed antigen which is being sensed by the T lymphocyte by two type of sensors of T lymphocyte. One is your TCR sensor, another is your MSC class 2 sensor, and MSC class 2 sensor is nothing, it will sense that whether it has been properly processed by MSC class 2 protein or not. So why I am saying all these things? Because a very important question asked from in the examination from this point. So what I told here, this is a processed antigen. This processed antigen is presenting itself to the T lymphocyte, right? This one is your T lymphocyte. This is T lymphocyte. This is T lymphocyte. So this processed antigen is presenting itself to the T lymphocyte for next reactions. So that's why it is called as antigen presenting cell. So the name of the justification of the name of the antigen presenting cell, now you are getting, I think, hope you have understood. What is antigen presenting cell? APC, as macrophage is presenting the processed antigen processed with the MSC class 2 protein to the T lymphocytes. That's why it is called as antigen presenting cell. It is a very important question asked in the examination. That what is antigen presenting cell? Antigen presenting cell is nothing but macrophage processed antigen which is processed by MSC class 2 protein, and that is presented to the T lymphocytes. That's why it is called as antigen presenting cell. So now let me put the answer in the chat box. Antigen presenting cell is related to which one? Neutrophil, macrophage, lymphocyte, basophil. You can answer, I think. Okay. So antigen presenting cell. Now you answer me another question, that macrophage was your what? Macrophage was a cell, and this is T, as the processed antigen is presented to the T lymphocytes, and that's why a question frequently asked in your examination, that cell mediated immunity is maintained by this kind of process. What is cell mediated immunity? You see, this is a T lymphocyte, this is macrophage, this is macrophage, this is macrophage. Macrophage presented the processed antigen to the T lymphocyte, right? Then immunity started over here, as macrophage is presenting the processed antigen to the T lymphocyte. So that's why cell mediated immunity is maintained by the T lymphocyte. You got my point? You got my point? In BHU it was asked, and also in many exams it is asked, that cell mediated immunity is maintained by, in many, in many exams it is called, get the question, you will get the question, cell mediated immunity. Cell mediated immunity Cell mediated immunity by what? T lymphocyte. Cell mediated immunity by what? T lymphocyte, my dear friends. Why T lymphocytes? Cell mediated immunity is by T lymphocyte. Why T lymphocyte? Now C for cell, that is for macrophage, which processed the antigen, then present into the T cell. So that's why cell mediated immunity is by T lymphocyte. It is very easy. Cell mediated immunity is by T cell. This is processed antigen presented to the T lymphocyte. That's why it is cell mediated immunity by the T lymphocytes. You got my point? Hope you have understood what I told. Okay, then another 2 minutes of things is there. Then what? Then cell mediated immunity and T lymphocytes will express its genetic material. Then what will happen? Then these T lymphocytes gene expression. Then from the T lymphocyte there will be gene expression. Then from the T lymphocyte there will be gene expression, and it will act upon what? Very interesting. T lymphocyte will act on what? Yes, please. T lymphocyte act on what? B lymphocyte. B lymphocyte from what? Bone marrow. T lymphocyte from what? Thymus gland. Entrance में आने वाला question. T lymphocyte from thymus gland. B lymphocyte from bone marrow. Then B lymphocyte will form what? Then B lymphocyte will form what? Finally, your antibodies. Then B lymphocyte will finally form what? Antibodies. It is very interesting, you see. B lymphocyte will form what? Antibodies. And these antibodies will act upon the bacteria, and these antibodies will now act upon these antibody will be formed, and these antibody will act against the bacteria. So now you say that how many questions you got from the entrance point of view questions. That then from the B lymphocytes another cell is there, that is plasma cell. Then plasma cell, cartwheel type of cell. From the plasma cells antibodies are formed. So many things are coming from here. Now you see that how much important your monocyte is. So in neutrophil all these things are happening. I have already told. Neutrophil, lysosomal enzymes are secreting, then killing the bacteria, then releasing the now antigens are free. Then these antigens are drained to the lymphatic system, then coming to the lymphoid headquarter, then to the lymph nodes, then it will be presented to the lymph nodes, thymus and lymphocytes. But in case of monocyte it is not happening here. So how many things, I will just recap it, how many things we learned from here. Then when monocyte is working, in the monocyte same thing is happening. From the monocyte lysosomal enzymes will damage the bacteria, then antigens will be made free, then these antigens will be made free, then inside the monocyte, inside the macrophage, again ignore the things, monocyte when inside the blood vessel, macrophage when outside the blood vessel. Then macrophage now in the chromosome number 6 in both paternal and maternal it is expressing some genetic material. So due to the genetic expression from DNA, you mean genetic expression, we are getting the mRNA, messenger RNA, and I am zoom it, and one part is a suppling part, next part is a stable part. From the DNA mRNA is formed, and mRNA you see first mRNA, second mRNA, third mRNA. Then this kind of proteins are formed, and these are thousands in number. Then these proteins will now select that which protein will be fitted to the antigen free, made free by the macrophage. Then now we got that this was selected and fitted to the antigen. The next what happened? This antigen this antigen now selected, and this antigen was presented to the selected T lymphocytes. So that's why it is called as processed antigen, as it was presented to the T lymphocytes. So that's what it is called as antigen presenting cell. So antigen presenting cell is related to macrophage. Then when antigen presenting cell is presenting it to the T lymphocyte, that means T lymphocyte is mediated by cell mediated immunity. Cell mediated immunity is mediated by T lymphocyte. It's a cell, It is T lymphocyte cell-mediated immunity. Then again, when T lymphocyte is getting the processed antigen, then T lymphocyte will sense by both of the things. One is with a two sensor. One is TCR, T lymphocyte receptor sensor. Another is a major histocompatibility class 2 sensor. Then whether they will sense that this processed antigen has been properly processed or not. Then after that, from the T lymphocyte, there will generate expression to the B lymphocyte. Then B lymphocyte, you know, it is from bone marrow, T lymphocyte from thymus gland. I asked in the entrance examinations. Then from the B lymphocytes, plasma cells, it is cartwheel type of cell. Then antibodies will form. Then these antibodies will be act on the next bacteria. So you mean what is happening here? That monocyte macrophage is doing such a big event that is again producing more number of antibodies, and these antibodies are responsible to kill the multiple number of bacteria over here. So now let me recap again one thing. Neutrophil, first line of defense mechanism, and asked in question examinations, why neutrophil is first line of defense mechanism? Because it is more in number, number one. Number two, neutrophil is more motile. Then number three, the first chemotactic agent secreted from the active site, infection site, so that neutrophils are getting attracted. So second line of defense mechanism, monocyte. When monocyte inside the blood vessels, it is monocyte. When it is coming out of the blood vessel, it is called as macrophage. Then monocyte, then neutrophil is having multi-lobed nucleus, and neutrophil is otherwise called as microphage. Then monocyte is called as macrophage. Monocyte is called as big eater. Monocyte has what kidney shaped nucleus. So many things I have told about the monocyte, macrocyte. Then a processed antigen is happening here. That's why it is called as antigen presenting cell. Then antigen presenting cell is again to the T lymphocytes. So that's why cell mediated immunity is maintained by the T lymphocytes. It is very frequently asked in the examination. Then T lymphocytes is again stimulating B lymphocyte. From B lymphocytes, plasma cells. Then plasma cells to antibodies are formed. Again these antibodies will come to the active site to kill the bacteria. Then apart from this, a complementary protein systems are there, that more than 20 numbers of complementary proteins are released by the liver, and by the help of the complementary proteins, again different kind of defense mechanisms happens inside our body. So these are the things I discussed over here. Why? Because these are from entrance point of view very interesting, very important, and everything on it. So thank you all for listening, for your patience. Note it down what I have told, and if you have any question, kindly put it in your WhatsApp or convey me, then I will answer you everything. So again, in my last class I am saying that have patience in spite of your busy schedule, is just keep concentrating in noting what I am saying, and I am assuring you that definitely you will do the best in your PG entrance examination, and also in Odisha Public Service Commission and Research Officer CCRAS all examinations. Thank you all, thank you. Bye bye, good night.